Every atom in your body was forged in the heart of a dying star, and every cell carries within it the unbroken thread of a story that began nearly 4 billion years ago. In darkness, in silence, in water so deep that sunlight had never touched it. The Earth in its infancy was a world unrecognizable to us now, a planet of violence and extremes, bombarded by asteroids, racked by volcanic eruptions that split the crust and poured molten rock across the surface. The atmosphere was thick with methane, ammonia, carbon dioxide and water vapor, but no oxygen, no breathable air as we know it. The oceans were hot, acidic, rich in dissolved minerals leached from the rocks, and it was in these oceans, in the darkness near hydrothermal vents on the seafloor, that the first spark of life may have ignited. These vents, fissures in the ocean floor, where superheated water rich in hydrogen, methane, and minerals erupts from the Earth's interior, create chemical gradients, differences in pH and temperature and composition, and these gradients can drive chemical reactions, can provide energy, can create the conditions necessary for the spontaneous assembly of complex organic molecules from simple precursors. The first cells, if we can call them that, were likely little more than membranes enclosing a few self-replicating molecules, but they were alive, capable of growth, of reproduction, of evolution, and from these humble beginnings all life on Earth descends. The chemistry of the early ocean was fundamentally different from the ocean we know today. Iron dissolved in the water gave it a greenish tint, and the absence of oxygen meant that reactions that would be impossible in the modern ocean proceeded freely, spontaneously, driven by the energy of the vents, by the ultraviolet radiation that penetrated the thin atmosphere, by the lightning that crackled through the methane-rich air. In this primordial soup, simple molecules combined to form more complex ones, amino acids linked together to form short chains called peptides, nucleotides linked together to form short strands of RNA, lipids assembled into vesicles, tiny bubbles of membrane that could encapsulate other molecules, protecting them from the environment, concentrating them, creating microenvironments where reactions could proceed more efficiently. These vesicles were not alive, not yet, but they were protocells, precursors to cells, and in some of them, by chance, by the random assembly of molecules, self-replicating systems emerged, systems that could make copies of themselves, that could grow, that could divide. And these systems were the first living things, the first organisms, the ancestors of all life that would follow. The transition from non-living chemistry to living biology is one of the deepest mysteries in science, a boundary so fundamental that we still do not fully understand how it was crossed. We know that life requires certain ingredients: liquid water, a source of energy, and organic molecules, the carbon-based compounds that form the building blocks of proteins, nucleic acids, lipids, and carbohydrates. We know that these ingredients were present on the early Earth, and we know that given the right conditions, organic molecules can form spontaneously from simpler compounds. The Miller-Urey experiment, conducted in 1953, demonstrated that amino acids, the building blocks of proteins, can be synthesized from a mixture of methane, ammonia, hydrogen, and water vapor, subjected to electrical sparks simulating lightning. Since then, similar experiments have produced nucleotides, the building blocks of DNA and RNA, and lipids, the molecules that form cell membranes. The chemistry of life is not magical, it is chemistry, governed by the same laws that govern all chemical reactions. But the step from chemistry to life, from molecules to metabolism, from reaction to replication, remains elusive, a gap in our understanding that we are working to close. One of the leading hypotheses for the origin of life is the RNA world hypothesis, which proposes that the first self-replicating molecules were not proteins or DNA, but RNA, ribonucleic acid, a molecule that can both store genetic information like DNA and catalyze chemical reactions like proteins. RNA is simpler than DNA, requiring fewer types of building blocks, and it is more versatile than proteins, capable of folding into complex three-dimensional shapes that can bind to other molecules and facilitate reactions. In the RNA world, RNA molecules would have replicated themselves using other RNA molecules as templates, and they would have catalyzed the reactions necessary to build more RNA, creating a self-sustaining cycle of replication and metabolism. Over time, some RNA molecules evolved the ability to catalyze the synthesis of proteins, which are more efficient catalysts than RNA, and eventually, the genetic information stored in RNA was transferred to DNA, which is more stable and less prone to mutation. The RNA world, if it existed, was a transitional stage, a stepping stone between non-living chemistry and the DNA protein world of modern cells. And traces of this ancient world persist in modern biology in the ribosomes that synthesize proteins, which are made largely of RNA, and in the use of RNA molecules as messengers carrying genetic information from DNA to the protein synthesis machinery. The concept of deep time, of billions of years, is almost impossible for the human mind to grasp. If we compress the entire history of the Earth into a single 24-hour day, life would appear around 4 a.m, in the darkness before dawn, and for the next 16 hours, for 2/3 of the day, life would consist only of single-celled organisms, invisible, silent, yet transforming the planet. Multicellular life would not appear until 8 in the evening, and humans would arrive only in the last second before midnight, a fleeting presence in a story that spans eons. This perspective, this sense of our place in deep time, is humbling, and it is essential for understanding evolution, for understanding that the processes we observe today, the mutations, the selection, the adaptation, have been operating for billions of years, accumulating changes so slowly that they are imperceptible in a human lifetime, but so relentlessly that they have transformed single-celled organisms into the vast diversity of life we see today. The earliest evidence of life on Earth comes from fossils, but not fossils of bones or shells which did not exist in the early Earth. The earliest fossils are stromatolites, layered structures formed by communities of microorganisms, primarily cyanobacteria, which trap and bind sediment, building up layers over time. Stromatolites are still forming today in a few locations around the world, in shallow, warm, salty waters where conditions resemble those of the early Earth, and they provide a window into the deep past, a glimpse of what life was like billions of years ago. The oldest stromatolites found in Western Australia and Greenland are approximately 3.5 billion years old, and they indicate that by that time life was not only present, but abundant, forming complex communities, photosynthesizing, producing oxygen, shaping the environment. But life must have originated earlier, perhaps as early as 4 billion years ago, shortly after the Earth's surface cooled enough for liquid water to exist, in the brief interval between the end of the heavy bombardment and the formation of the first stromatolites, a period of perhaps 500 million years. A geological eye blink, but long enough for chemistry to become biology, for molecules to become cells, for the first spark of life to ignite and spread. The early cells were prokaryotes, simple cells without a nucleus, without membrane-bound organelles, resembling modern bacteria and archaea. They were small, typically only a few micrometers in diameter, and they were metabolically diverse, capable of extracting energy from a wide range of chemical reactions, from the oxidation of hydrogen and methane near hydrothermal vents, to the reduction of sulfur and iron in the water column, to the splitting of water molecules using the energy of sunlight, a process called photosynthesis. The evolution of photosynthesis, specifically oxygenic photosynthesis, in which water is split and oxygen is released as a byproduct, was one of the most important events in the history of life because it transformed the atmosphere, filling it with oxygen, a highly reactive gas that was toxic to most of the organisms that existed at the time. The Great Oxidation Event, which occurred approximately 2.5 billion years ago, was a catastrophe for anaerobic life, for the organisms that thrived in the absence of oxygen. But it was also an opportunity. Because oxygen enables aerobic respiration, a metabolic pathway that extracts far more energy from organic molecules than anaerobic pathways. And aerobic respiration made possible the evolution of larger, more complex, more energetically expensive forms of life. The accumulation of oxygen in the atmosphere was not instantaneous. For hundreds of millions of years, the oxygen produced by cyanobacteria was consumed by reactions with iron and other reduced minerals in the ocean and on land, forming rust-colored deposits, the banded iron formations that are mined today for their iron content. Only when these oxygen sinks were saturated, when there was no more reduced iron to oxidize, did oxygen begin to accumulate in the atmosphere, rising from trace levels to significant concentrations, eventually reaching levels high enough to form an ozone layer in the upper atmosphere, which shields the surface from harmful ultraviolet radiation. This ozone layer made it possible for life to colonize the land, to move out of the protective cover of water and into the harsh environment of the terrestrial surface. But that colonization would not occur for another billion years, during which time life remained aquatic, evolving, diversifying, preparing for the next great transition. The origin of eukaryotic cells, cells with a nucleus and membrane-bound organelles, is another major transition in the history of life, and it occurred through a process called endosymbiosis, in which one cell engulfs another, and instead of digesting it, the two cells form a partnership, a mutualism in which both benefit. The mitochondria, the organelles that perform aerobic respiration in eukaryotic cells, were once free-living bacteria that were engulfed by a larger cell, and over time they lost their independence, transferring most of their genes to the nucleus of the host cell, becoming integrated into the host's metabolism, becoming essential. The chloroplasts, the organelles that perform photosynthesis in plant cells and algae, have a similar origin. They were once free-living cyanobacteria that were engulfed by a eukaryotic cell, and they too became integrated, became essential, became organelles. The evidence for endosymbiosis is overwhelming. Mitochondria and chloroplasts have their own DNA, distinct from the nuclear DNA, and this DNA is circular, like bacterial DNA, not linear like eukaryotic DNA. They have their own ribosomes, which are more similar to bacterial ribosomes than to eukaryotic ribosomes. They reproduce by binary fission, like bacteria, not by mitosis, like eukaryotic cells. And they are surrounded by two membranes, the inner membrane derived from the original bacterial cell, and the outer membrane derived from the host cell's membrane that engulfed them. Endosymbiosis was not a single event. It happened multiple times in different lineages, and it was a key innovation that enabled the evolution of complex multicellular life, because mitochondria provide the energy necessary to support the metabolic demands of large active organisms. The deep time of the early Earth, the billions of years during which life was exclusively microbial, is difficult for us to comprehend. We are accustomed to thinking of history in terms of human lifespans of decades and centuries, but the history of life is measured in billions of years, in timescales so vast that they defy intuition. For more than half of Earth's history, for more than 2 billion years, life consisted only of single-celled organisms, bacteria and archaea, invisible to the naked eye, yet profoundly shaping the planet, creating the oxygen in the atmosphere, depositing the iron in the rocks, forming the stromatolites that are among the oldest fossils we have. These organisms were not simple. They were metabolically sophisticated, genetically diverse, ecologically complex, forming communities, exchanging genes, evolving, adapting, surviving mass extinctions and climate shifts and the slow drift of continents. They were the pioneers, the foundation, the ancestors of all life that came after, and they persist today in the soil, in the oceans, in the air, in our bodies, outnumbering all other forms of life, essential to the functioning of ecosystems, to the cycling of nutrients, to the health of the planet. The genetic code, the system by which sequences of nucleotides in DNA are translated into sequences of amino acids in proteins, is universal, shared by all life on Earth, from bacteria to humans. This universality is one of the strongest pieces of evidence for common ancestry, for the idea that all life descends from a single origin, a single population of organisms that lived billions of years ago. The code is not random. Certain patterns suggest optimization, efficiency, error correction, but it is also arbitrary in many respects, and the fact that all organisms use the same arbitrary code indicates that they inherited it from a common ancestor. There are slight variations in the genetic code in some organisms, in mitochondria, and in some bacteria, but these variations are minor, and they occurred after the main branches of the tree of life had diverged. The core of the code, the fundamental mapping of nucleotide triplets to amino acids, is ancient, unchanged for billions of years, a molecular fossil, a trace of the earliest life, preserved in every cell, in every genome, in every organism alive today. The first spark of life, ignited in the darkness of the deep ocean, near a hydrothermal vent, or perhaps in a shallow tidal pool, or in a film of water on the surface of a mineral grain, was a fragile thing, a chemical accident, a self-replicating molecule that happened to persist, to reproduce, to evolve. But from that fragile beginning, from that single spark, all the diversity of life on Earth has emerged through billions of years of evolution, of mutation and selection, of adaptation and innovation, of extinction and radiation. Every organism alive today, from the smallest bacterium to the largest whale, from the simplest virus to the most complex human, shares a common ancestor, a common origin, a common chemistry, a common genetic code. We are all related, all connected, all part of the same unbroken lineage, the same tree of life, rooted in the deep past, branching into the present, reaching toward the future. And in our cells, in our DNA, in the mitochondria that power our metabolism, we carry the traces of that ancient origin, the echoes of the first spark, the memory of the abyss, the silence of deep time, the patient unfolding of 4 billion years of evolution, written in the language of molecules, in the sequences of genes, in the structures of proteins, in the rhythms of metabolism, in the very fact of our existence, which is a testament to the power of life to emerge, to endure, to adapt, to persist against all odds across unimaginable stretches of time in a universe that is vast, cold, and indifferent, but which on this small blue planet has given rise to something extraordinary, something precious, something worth protecting, worth understanding, worth marveling at, in silence, in wonder, in the deep peace of knowing that we are part of something far larger, far older, far more profound than ourselves. The architecture of life is not random. Every vertebrate that has ever lived, from the first fish to the last human, shares the same basic body plan, the same arrangement of bones, the same genetic toolkit, variations on a theme written 400 million years ago, and never forgotten. When you raise your hand, you are using bones that first appeared in the fins of ancient fish, bones that have been modified, reshaped, repurposed, but never discarded, never redesigned from scratch. The humerus in your upper arm, the radius and ulna in your forearm, the carpals in your wrist, the metacarpals in your palm, the phalanges in your fingers. These same bones, in the same order, appear in the wing of a bat, the flipper of a whale, the leg of a horse, the paw of a cat. The proportions differ, the functions differ, but the underlying structure is identical, a pattern inherited from a common ancestor, a lobe-finned fish that lived in the shallow waters of the Devonian period, approximately 375 million years ago. This is homology, the similarity of structure due to shared ancestry, and it is one of the most powerful pieces of evidence for evolution, for the idea that all vertebrates are related, that they share a common origin, that they have diverged from a single ancestral form through the process of descent with modification. The pentadactyl limb, the five-fingered limb, is not the optimal design for every function. A horse would run faster with a single strong digit, and indeed, over millions of years, horses have reduced their 5 toes to one, the middle toe, which bears the weight, while the other toes have become vestigial, reduced to small bones called splints that serve no function. A whale does not need fingers at all, and yet inside the flipper of a whale, there are 5 digits, clearly visible in the skeleton, encased in flesh and blubber, a remnant of the whale's terrestrial ancestry, a trace of the time when the ancestors of whales walked on land. Evolution does not design from scratch. It modifies what already exists, tinkering with existing structures, repurposing them for new functions, constrained by history, by the genetic and developmental pathways that have been inherited from ancestors. This is why we see the same basic patterns repeated across diverse groups of organisms, why the forelimbs of all tetrapods, all four-limbed vertebrates, share the same skeletal structure despite being used for flying, swimming, running, grasping, digging, climbing, functions that seem to have nothing in common. The genetic basis of development reveals even deeper homologies, similarities that are not visible in the adult form, but are evident in the genes that control the formation of the body during embryonic development. The Hox genes, a family of genes that specify the identity of body segments along the head-to-tail axis, are found in all animals from flies to humans, and they are arranged in the same order on the chromosome, and they are expressed in the same order along the body axis, from head to tail. A mutation in a Hox gene in a fruit fly can cause legs to grow where antennae should be, or wings to grow where legs should be, demonstrating that these genes are master regulators, controlling the development of entire body parts. The fact that humans and flies share the same Hox genes with similar functions indicates that these genes were present in the common ancestor of all animals, an ancestor that lived more than 600 million years ago, and they have been preserved by natural selection, because they perform essential functions that cannot be easily replaced. The eye, often cited as an example of irreducible complexity, of a structure so intricate that it could not have evolved gradually, is actually a powerful example of homology and convergent evolution. The camera-type eye of vertebrates, with a lens that focuses light onto a retina, appears to be fundamentally different from the compound eye of insects, with hundreds or thousands of individual lenses, each forming a separate image. But at the genetic level, the development of both types of eyes is controlled by the same gene, called PAX6 in vertebrates and eyeless in fruit flies. If you take the PAX6 gene from a mouse and insert it into a fruit fly, you can induce the formation of an eye, a fly eye, not a mouse eye, on the fly's wing or leg or antenna, demonstrating that PAX6 is a master control gene that triggers the entire developmental program for eye formation, and that this gene was present in the common ancestor of vertebrates and insects, an ancestor that lived more than 500 million years ago. The eyes themselves evolved independently through different pathways, using different structures, but the genetic switch that initiates eye development is the same, a shared inheritance from a distant ancestor. The embryonic development of vertebrates provides another window into evolutionary history. All vertebrate embryos, whether they will develop into fish, amphibians, reptiles, birds, or mammals, pass through similar stages of development, and at certain stages, they are almost indistinguishable from one another. Human embryos, at around 4 weeks of development, have gill slits, structures that in fish develop into gills, the organs used for breathing underwater. In humans, these gill slits do not develop into gills. They are remodeled, transformed into other structures: the bones of the middle ear, the thymus gland, the parathyroid glands, structures that have nothing to do with breathing underwater. But the fact that human embryos have gill slits at all is evidence of our evolutionary history, evidence that our ancestors were fish, that we carry within our developmental program the traces of that aquatic past, traces that are expressed during embryonic development and then modified, repurposed, transformed into structures suited for life on land. The recurrent laryngeal nerve in mammals is another example of evolutionary constraint, of a structure that makes no sense from a design perspective, but makes perfect sense from an evolutionary perspective. This nerve, which controls the muscles of the larynx, the voice box, takes a circuitous route from the brain down into the chest, looping around the aorta, the main artery leaving the heart, and then travelling back up to the larynx. In humans, this detour adds a few inches to the length of the nerve, an inefficiency but not a major problem. In giraffes, however, the recurrent laryngeal nerve travels all the way down the neck, loops around the aorta in the chest, and then travels all the way back up the neck to the larynx, a detour of more than 15 feet. An absurd inefficiency. The reason for this detour is evolutionary history. In fish, the ancestors of all tetrapods, the nerve takes a direct route from the brain to the gills, passing behind the aortic arch. When tetrapods evolved and the neck elongated, the nerve was already committed to this route, constrained by the developmental pathways inherited from fish. And as the neck grew longer, the nerve grew longer, maintaining its connection to the aorta, unable to take a more direct route because that would require a complete rewiring of the developmental program, a change so drastic that it has never occurred. Evolution is not an engineer designing from scratch. It is a tinkerer modifying existing structures, constrained by history, by the pathways that have been inherited from ancestors. The molecular clock, the observation that mutations accumulate in DNA at a relatively constant rate over time, allows us to estimate the time since two species diverged from a common ancestor by comparing the differences in their DNA sequences. The more differences, the longer the time since divergence. This molecular clock has been calibrated using the fossil record, using known divergence times for species that have left fossils, and it has been used to estimate divergence times for species that have not left fossils, or for which the fossil record is incomplete. The molecular clock is not perfectly regular. The rate of mutation varies among different genes, among different lineages, among different time periods, but on average, over long timescales, it provides a reasonably accurate estimate of evolutionary time. Using the molecular clock, we can estimate that humans and chimpanzees diverged from a common ancestor approximately 6 to 7 million years ago, that humans and mice diverged approximately 90 million years ago, that humans and fish diverged approximately 450 million years ago, and that all animals diverged from a common ancestor approximately 600 to 700 million years ago. The universality of the genetic code, the fact that all organisms use the same code to translate DNA sequences into protein sequences, is powerful evidence for common ancestry. There are 64 possible triplets of nucleotides called codons, and each codon specifies one of 20 amino acids, or a stop signal indicating the end of a protein. The assignment of codons to amino acids is arbitrary. There is no chemical reason why a particular codon should specify a particular amino acid, and yet all organisms, from bacteria to humans, use the same assignments, with only minor variations in a few organisms. This universality indicates that the genetic code was established very early in the history of life, in the common ancestor of all organisms, and it has been conserved ever since because any change to the code would be catastrophic, altering the structure of every protein in the organism, almost certainly resulting in death. The genetic code is a frozen accident, a historical contingency that has become universal, a shared inheritance that links all life on Earth. The distribution of species across the globe provides additional evidence for evolution and common ancestry. Species that are closely related tend to be found in the same geographic regions, even when those regions are separated by barriers like oceans or mountains. The marsupials, mammals that give birth to underdeveloped young that complete their development in a pouch, are found primarily in Australia and South America, regions that were once connected as part of the supercontinent Gondwana. When Gondwana broke apart, approximately 180 million years ago, the marsupials on the Australian fragment were isolated from the marsupials on the South American fragment, and they evolved independently, diversifying into different forms, but retaining the basic marsupial characteristics. The fact that marsupials are found in Australia and South America, but not in Africa or Asia or North America, except for the opossum, which migrated from South America relatively recently, is evidence that they evolved from a common ancestor that lived on Gondwana before it broke apart, and that their current distribution reflects the history of continental drift, the slow movement of continents across the surface of the Earth. Island biogeography, the study of species on islands, provides some of the clearest evidence for evolution. Islands, especially oceanic islands that have never been connected to continents, have fewer species than continents, and the species they do have are often unique, found nowhere else in the world. The Galapagos Islands, located in the Pacific Ocean about 600 miles off the coast of Ecuador, are home to species of finches, tortoises, iguanas, and mockingbirds that are found nowhere else, and these species are closely related to species on the South American mainland, indicating that they descended from mainland ancestors that colonized the islands, probably by flying or floating on vegetation, and then evolved in isolation, adapting to the unique conditions of the islands, diversifying into new forms. The finches of the Galapagos, made famous by Charles Darwin, have diversified into more than a dozen species, each with a different beak shape adapted to a different food source, some eating seeds, some eating insects, some eating cactus flowers, some even drinking blood from seabirds. This adaptive radiation, the rapid diversification of a single ancestral species into multiple species occupying different ecological niches, is a hallmark of evolution on islands, where competition is low and opportunities are abundant. The fossil record, though incomplete, provides direct evidence of evolutionary change over time, showing the appearance of new forms, the modification of existing forms, and the extinction of old forms. Transitional fossils, fossils that show intermediate characteristics between two groups of organisms, are particularly important because they demonstrate that one group evolved from another, that the transition was gradual, not sudden. Tiktaalik, a fossil discovered in Arctic Canada in 2004, is a transitional form between fish and tetrapods, four-limbed vertebrates. Tiktaalik lived approximately 375 million years ago, and it had features of both fish and tetrapods. It had scales, gills, and fins like a fish, but it also had a flat head, a neck, and sturdy ribs like a tetrapod, and its fins had bones that correspond to the humerus, radius, and ulna of tetrapod limbs, suggesting that it could support its weight on its fins and perhaps even walk on the bottom of shallow streams. Tiktaalik is not the direct ancestor of all tetrapods, but it is a close relative, a representative of the group from which tetrapods evolved, and it provides a glimpse of what the transition from water to land looked like, a transition that was gradual, taking millions of years, involving many intermediate forms, most of which have not been preserved in the fossil record. The branching tree of life, the phylogenetic tree that shows the evolutionary relationships among all organisms, is constructed from multiple lines of evidence: comparative anatomy, embryology, molecular biology, biogeography, and paleontology. All of these lines of evidence converge on the same tree, the same pattern of relationships, indicating that evolution is not a hypothesis but a fact, a process that has shaped the diversity of life on Earth, a process that continues today in every population, in every generation, driven by mutation, selection, drift, and migration. The forces that have been operating for billions of years and will continue to operate for billions more, as long as life persists, as long as the sun shines, as long as the earth endures. Your body is a museum of evolutionary history, carrying within it structures that no longer serve their original purpose, remnants of functions that were essential to your ancestors but have become obsolete, footnotes written in flesh and bone that tell the story of where you came from. The appendix, a small finger-shaped pouch attached to the large intestine, has long been considered a vestigial organ, a remnant of a larger cecum that was used by herbivorous ancestors to digest cellulose, the tough plant material that makes up the cell walls of plants. In animals like rabbits and horses, the cecum is large and houses bacteria that break down cellulose, extracting nutrients that the animal cannot access on its own. In humans, the cecum is much smaller, and the appendix is even smaller, a narrow tube that serves no obvious digestive function and is prone to inflammation, a condition called appendicitis that can be life-threatening if not treated. For many years, the appendix was thought to be completely useless, a vestige of our herbivorous past, but recent research suggests that it may serve as a reservoir for beneficial gut bacteria, a safe house where bacteria can survive when the main population in the intestine is depleted by disease or antibiotics, and from which they can recolonize the gut after the threat has passed. Whether or not the appendix serves this function in modern humans, its structure and position are evidence of evolutionary history, of a time when our ancestors had a larger cecum and a more plant-based diet. The coccyx, the tailbone, is another vestigial structure, a remnant of the tail that our ancestors possessed. In human embryos, at around 4 to 5 weeks of development, there is a visible tail, complete with vertebrae, extending from the base of the spine. As development proceeds, this tail is reabsorbed, and by birth, only a small remnant remains, the coccyx, consisting of 3 to 5 fused vertebrae. The coccyx serves as an attachment point for muscles and ligaments involved in pelvic floor support, but it is not essential, and people who have their coccyx surgically removed due to injury or chronic pain experience no significant loss of function. The coccyx is a trace of our evolutionary past, evidence that our ancestors had tails, as do most mammals, and that we have lost that tail over millions of years of evolution, as our ancestors adapted to bipedal locomotion and life in the trees, where a tail was less useful than in quadrupedal animals that use their tails for balance or communication. Wisdom teeth, the third molars that typically erupt in late adolescence or early adulthood, are another example of vestigial structures. In our ancestors, who had larger jaws and a diet that consisted of tough, fibrous plant material and raw meat, wisdom teeth were necessary for grinding food, and the jaw was large enough to accommodate them. In modern humans, jaws have become smaller, partly due to changes in diet, particularly the shift to cooked and processed foods that require less chewing, and partly due to genetic changes that have occurred over thousands of years. As a result, many people do not have enough room in their jaws for wisdom teeth, and when these teeth erupt, they become impacted, trapped beneath the gum, or growing at odd angles, causing pain, infection, and damage to adjacent teeth. Wisdom teeth are often removed surgically, and their absence causes no problems, indicating that they are no longer necessary, that they are vestiges of a time when our ancestors had larger jaws and a different diet. The muscles that move the ears, the auricular muscles, are present in humans but are largely non-functional. In many mammals, these muscles are well developed and allow the animal to move its ears independently, orienting them towards sounds, a useful ability for detecting predators or prey. In humans, these muscles are small and weak, and most people cannot move their ears voluntarily, though a few can wiggle them slightly, a party trick with no practical value. The presence of these muscles and the nerves that innervate them is evidence that our ancestors could move their ears, and that this ability has been lost over time as our ancestors became more reliant on vision and less reliant on hearing for detecting threats and opportunities. The plica semilunaris, a small fold of tissue in the inner corner of the eye, is a remnant of the nictitating membrane, the third eyelid that is present in many animals, including birds, reptiles, and some mammals. The nictitating membrane can move horizontally across the eye, protecting it from debris and moisture, while still allowing the animal to see. In humans, the nictitating membrane has been reduced to a small, non-functional fold, the plica semilunaris, which serves no purpose but remains as a trace of our evolutionary history, evidence that our ancestors had a third eyelid and that we have lost it over millions of years of evolution. Goosebumps, the small bumps that appear on the skin when we are cold or frightened, are another vestigial response, a remnant of a reflex that was useful in our hairier ancestors. When the tiny muscles at the base of each hair follicle contract, they cause the hair to stand upright, and in animals with thick fur, this creates a layer of insulation, trapping air close to the skin and reducing heat loss. In humans, who have relatively little body hair, goosebumps provide no insulation, no warmth, but the reflex persists, triggered by cold or by the adrenaline released during fear or excitement. The reflex may have served another function in our ancestors, making them appear larger and more threatening to predators or rivals. A display that is still effective in animals with fur, but is ineffective in hairless humans. Goosebumps are a trace of our evolutionary past, evidence that our ancestors were covered in fur, and that we have lost most of that fur over millions of years of evolution, as our ancestors adapted to warmer climates and developed other means of thermoregulation, such as sweating. The palmaris longus muscle, which runs from the elbow to the wrist, is present in about 85% of humans, but it is absent in about 15%, with no apparent disadvantage. This muscle is used by animals that climb or grasp branches, and it is well developed in primates that spend a lot of time in trees. In humans, the muscle is thin and weak, and it contributes little to grip strength or wrist function. Surgeons often harvest the palmaris longus tendon for use in reconstructive surgery because it can be removed without affecting hand function, demonstrating that it is not essential. The variability in the presence of this muscle and its lack of function in those who have it indicate that it is in the process of being lost, that it is a vestigial structure that was useful to our arboreal ancestors, but is no longer necessary in modern humans. The vomeronasal organ, also called Jacobson's organ, is a chemosensory structure found in many animals, particularly reptiles and mammals, that detects pheromones, chemical signals used for communication, especially in mating and territorial behavior. In humans, the vomeronasal organ is present during fetal development, but it regresses and becomes non-functional in most adults, though small pits in the nasal septum mark its location. The genes that encode the receptors for the vomeronasal organ are present in the human genome, but many of them are non-functional, pseudogenes that have accumulated mutations and no longer produce working proteins. The presence of the vomeronasal organ in human embryos and the presence of pseudogenes for its receptors in the human genome are evidence that our ancestors used this organ to detect pheromones, and that we have lost this ability over millions of years of evolution as our ancestors became more reliant on vision and less reliant on chemical communication. The erector pili muscles, the tiny muscles that cause goosebumps, are not the only vestigial muscles in the human body. The pyramidal muscle, a small triangular muscle in the lower abdomen, is present in about 80% of humans, but it is absent in about 20%. with no apparent effect on abdominal function. The plantaris muscle, a small muscle in the calf, is present in about 90% of humans, but it is absent in about 10%. And like the palmaris longus, it is often harvested for use in reconstructive surgery because it can be removed without affecting leg function. These muscles, and others like them, are remnants of our evolutionary past, structures that were useful to our ancestors, but have become less important or completely unnecessary in modern humans, and they're in the process of being lost, disappearing from the population through the accumulation of mutations that disrupt their development or function. The human genome itself is a record of evolutionary history, filled with pseudogenes, non-functional copies of genes that were once functional but have been inactivated by mutations. Humans have hundreds of olfactory receptor pseudogenes, genes that in other mammals encode receptors for detecting odors, but in humans, these genes have accumulated mutations that prevent them from producing functional proteins. The presence of these pseudogenes indicates that our ancestors had a much better sense of smell than we do, with hundreds of functional olfactory receptors, and that we have lost many of these receptors over millions of years of evolution. As our ancestors became more reliant on vision and less reliant on smell for finding food, detecting predators, and communicating with others, the pattern of pseudogenes in the human genome matches the pattern predicted by evolutionary theory. Genes that are no longer under selection, genes that are no longer necessary for survival or reproduction, accumulate mutations at a predictable rate, and over time they become non-functional, becoming pseudogenes, molecular fossils that record the history of gene loss and evolutionary change. The presence of vestigial structures in the human body and in the bodies of all organisms is powerful evidence for evolution, for the idea that organisms have descended from ancestors with different structures and different functions, and that evolution proceeds by modifying existing structures, not by designing new ones from scratch. Vestigial structures are not perfectly designed. They are the remnants of structures that were designed for different purposes, in different environments, in different bodies. And they persist because evolution is a historical process, constrained by the past. Unable to start over, unable to erase the traces of history, every vestigial structure is a footnote, a reminder that we are not the product of intelligent design, but the product of a long, slow, undirected process of descent with modification. A process that has shaped us and all living things, a process that continues today as our bodies continue to change, to adapt, to evolve, carrying forward the traces of the past into an uncertain future. The vestigial structures in our bodies are not evidence of poor design, they are evidence of history, of the deep time that separates us from our ancestors, of the countless generations that have lived and died, each one slightly different from the one before, each one shaped by the forces of mutation and selection, of drift and migration, of chance and necessity. These structures are reminders that we are part of the tree of life, that we are connected to all other organisms by threads of ancestry that stretch back billions of years, to the first cells, to the first spark of life in the ancient ocean. And in the silence of these vestigial structures, in the quiet persistence of these anatomical footnotes, there is a kind of peace, a recognition that we are not separate from nature but part of it, not above the process of evolution, but subject to it, shaped by it, defined by it, in ways both profound and subtle, in ways that are written in our bodies, in our genes, in the very structure of our cells, in the unbroken chain of life that links us to the past and carries us into the future. Evolution does not follow a single path. Across the vast distances of time and geography, separated by oceans and continents, unrelated organisms have arrived at the same solutions to the same problems, sculpting similar forms from different ancestral materials, proving that the laws of physics and the demands of survival constrain life in predictable ways. The wings of bats, birds, and pterosaurs are superficially similar, all adapted for powered flight, all shaped by the demands of aerodynamics, but they are constructed from different anatomical materials, evolved independently at different times in different lineages. The bird wing is supported by elongated arm bones and fused hand bones, with feathers extending from the arm and hand to create the airfoil. The bat wing is supported by elongated finger bones, with a thin membrane of skin stretched between the fingers, the arm, the body, and the hind legs. The pterosaur wing, in the extinct flying reptiles that dominated the skies during the age of dinosaurs, was supported by a single enormously elongated fourth finger, with a membrane stretched from that finger to the body and hind legs. All three wings perform the same function. All three are shaped by the same aerodynamic principles, but they are built from different structures, modified from the pentadactyl limb in different ways, demonstrating that evolution can arrive at the same solution through different routes, that there are multiple ways to build a wing, and that natural selection, acting on different starting materials, can produce similar outcomes. This is convergent evolution, the independent evolution of similar features in unrelated lineages, driven by similar selective pressures, by similar environmental challenges, by the universal constraints of physics and chemistry. The camera-type eye with a lens that focuses light onto a retina has evolved independently at least 6 times in vertebrates, in cephalopods like octopuses and squids, in some jellyfish, in some annelid worms, in some spiders, and in some crustaceans. The vertebrate eye and the cephalopod eye are remarkably similar in structure, both with a cornea, a lens, an iris, and a retina, but they differ in important details. In the vertebrate eye, the photoreceptor cells in the retina are oriented backward, with the light-sensitive part facing away from the incoming light, and the nerve fibers from these cells pass across the surface of the retina and converge to form the optic nerve, which exits through the retina, creating a blind spot where there are no photoreceptors. In the cephalopod eye, the photoreceptor cells are oriented forward, with the light-sensitive part facing the incoming light, and the nerve fibers pass behind the retina, so there is no blind spot. The vertebrate eye is wired backward, an inefficiency that is the result of evolutionary history, of the way the eye develops from an outgrowth of the brain, while the cephalopod eye is wired correctly, the result of a different developmental pathway. Despite these differences, the overall structure and function of the two eyes are similar, demonstrating that the camera-type eye is a good solution to the problem of forming images, and that natural selection, acting independently in different lineages, has converged on this solution multiple times. The streamlined body shape of dolphins, sharks, and ichthyosaurs, the extinct marine reptiles, is another example of convergent evolution. All three are fast-swimming predators, and all three have evolved a similar body shape, a fusiform body tapered at both ends, with a dorsal fin for stability, pectoral fins for steering, and a powerful tail for propulsion. Dolphins are mammals descended from terrestrial ancestors that returned to the ocean approximately 50 million years ago. Sharks are fish, with a lineage that extends back more than 400 million years. Ichthyosaurs were reptiles descended from terrestrial ancestors that returned to the ocean approximately 250 million years ago. Despite their different ancestries, despite the fact that they are built from different materials, dolphins have bones, sharks have cartilage. Ichthyosaurs had bones. They have converged on the same body shape, the same solution to the problem of moving efficiently through water, shaped by the demands of hydrodynamics, by the need to minimize drag and maximize thrust. The similarity is so striking that ichthyosaur fossils were initially mistaken for dolphin fossils, until closer examination revealed the differences in skeletal structure, in the arrangement of bones, in the presence of features like the sclerotic ring in the eye, which is present in reptiles but not in mammals. The evolution of C4 photosynthesis, a more efficient form of photosynthesis that is adapted to hot, dry environments, has occurred independently at least 60 times in different plant lineages. C4 plants, which include corn, sugarcane, and many tropical grasses, have a modified leaf anatomy and a modified biochemical pathway that concentrates carbon dioxide around the enzyme rubisco, reducing photorespiration and increasing water use efficiency. The evolution of C4 photosynthesis requires multiple changes in leaf anatomy, in enzyme expression, in the regulation of metabolic pathways, and yet it has evolved repeatedly in different families of plants in different parts of the world at different times, demonstrating that the selective pressure of hot, dry environments is strong enough to drive the evolution of this complex trait, and that there are multiple genetic pathways that can lead to the same outcome. The repeated evolution of C4 photosynthesis is evidence that evolution is not random, that it is shaped by the environment, by the challenges that organisms face, and that similar challenges can produce similar solutions, even in unrelated lineages. The evolution of echolocation, the ability to navigate and hunt using reflected sound waves, has occurred independently in bats and in toothed whales, including dolphins and porpoises. Both groups produce high-frequency sounds and listen for the echoes that bounce back from objects in the environment, using the timing and the intensity of the echoes to determine the location, the size, and the shape of objects. The evolution of echolocation requires changes in the vocal apparatus, in the auditory system, and in the brain regions that process sound, and yet it has evolved independently in these two groups, which are separated by more than 100 million years of evolutionary history. The genes involved in hearing, particularly the genes that encode proteins in the cochlea, the part of the inner ear that detects sound, show evidence of convergent evolution, with similar mutations occurring independently in bats and in whales, mutations that enhance the ability to detect high-frequency sounds, the frequencies used in echolocation. This molecular convergence, the independent evolution of similar genetic changes in response to similar selective pressures, is powerful evidence that evolution is not random, that it is shaped by the demands of the environment, and that similar demands can produce similar genetic solutions. The evolution of venom has occurred independently in many different lineages, in snakes, in spiders, in scorpions, in cone snails, in jellyfish, in some fish, in some mammals like the platypus and some shrews. Venom is a complex mixture of proteins and peptides that disrupt the physiology of prey or predators, causing paralysis, pain, tissue damage, or death. The evolution of venom requires the modification of existing genes, often genes that encode digestive enzymes or immune proteins, and the development of specialized glands and delivery systems like fangs or stingers. Despite the complexity of venom systems, they have evolved repeatedly in unrelated lineages, demonstrating that venom is an effective strategy for subduing prey or deterring predators, and that natural selection can favor the evolution of venom in any lineage that has the right starting materials and the right selective pressures. The evolution of bioluminescence, the ability to produce light through chemical reactions, has occurred independently in many different lineages, in bacteria, in fungi, in dinoflagellates, in jellyfish, in fireflies, in some fish, in some squid. Bioluminescence is used for a variety of purposes: attracting mates, luring prey, deterring predators, communicating with members of the same species, camouflaging the silhouette against downwelling light in the ocean. The chemical reactions that produce bioluminescence are similar across different groups, involving the oxidation of a substrate called luciferin by an enzyme called luciferase, but the specific luciferins and luciferases are different, indicating that bioluminescence has evolved independently, using different biochemical pathways in different lineages. The repeated evolution of bioluminescence is evidence that the ability to produce light is advantageous in many different environments, and that natural selection can favor the evolution of this ability in any lineage that can evolve the necessary biochemical machinery. The evolution of sociality, the tendency to live in groups and cooperate with others, has occurred independently in many different lineages. In insects like ants, bees and termites, in mammals like primates, elephants and whales, in birds like crows and parrots, in some fish, in some shrimp. Social living provides benefits such as improved defense against predators, more efficient foraging, and cooperative care of offspring. But it also imposes costs such as increased competition for resources and increased risk of disease transmission. The evolution of sociality requires changes in behavior, in communication, in the ability to recognize and remember individuals, and in some cases, in the willingness to sacrifice personal reproduction for the benefit of the group, as in the eusocial insects, where only the queen reproduces and the workers are sterile. Despite the complexity of social behavior, it has evolved repeatedly in unrelated lineages, demonstrating that the benefits of sociality can outweigh the costs in many different environments, and that natural selection can favor the evolution of cooperation, even in a world where competition is the norm. The evolution of viviparity, the retention of embryos inside the mother's body and the birth of live young, has occurred independently in many different lineages, in mammals, in some reptiles like some snakes and lizards, in some fish like sharks and guppies, in some amphibians, in some invertebrates like some scorpions and some insects. Viviparity provides benefits, such as protection of the embryos from predators and from environmental extremes, and the ability to provision the embryos with nutrients directly from the mother's body. But it also imposes costs, such as the energetic burden on the mother and the reduction in the number of offspring that can be produced at one time. Despite these costs, viviparity has evolved repeatedly in unrelated lineages, demonstrating that the benefits can outweigh the costs in many different environments, and that natural selection can favor the evolution of live birth, even in groups that ancestrally laid eggs. Convergent evolution is not limited to morphology and physiology. It also occurs at the molecular level, in the sequences of genes and proteins. The evolution of antifreeze proteins, which prevent ice crystals from forming in the blood and tissues of organisms living in freezing environments, has occurred independently in Arctic fish and in Antarctic fish, which are separated by thousands of miles and millions of years of evolutionary history. The antifreeze proteins in these two groups are structurally different, encoded by different genes, but they perform the same function, binding to ice crystals and preventing them from growing, allowing the fish to survive in water that is below the freezing point of normal blood. The independent evolution of antifreeze proteins in Arctic and Antarctic fish is evidence that the selective pressure of freezing temperatures is strong enough to drive the evolution of this adaptation, and that there are multiple genetic solutions to the problem of surviving in the cold. The patterns of convergent evolution reveal the constraints and the possibilities of life, the ways in which the laws of physics and chemistry and the demands of survival shape the diversity of organisms. Convergent evolution shows that evolution is not entirely random, that it is channeled by the environment, by the challenges that organisms face, and that similar challenges can produce similar solutions even in lineages that are separated by vast distances of time and space. And in the silence of these convergent forms, in the repeated invention of wings and eyes and streamlined bodies, there is a kind of order, a recognition that life, for all its diversity, is governed by universal principles, by the same forces that shape the stars and the galaxies, by the same laws that have been operating since the beginning of time, and that will continue to operate, shaping life, shaping evolution, shaping the future as long as the universe endures. The journey from water to land was not a single step, but a series of transformations spanning millions of years, a slow migration from the buoyant silence of the ocean to the harsh exposure of air, requiring the reinvention of nearly every system in the body, From breathing to reproduction to the very structure of the skeleton, the first vertebrates were fish, creatures born in water, shaped by water, entirely dependent on water for every aspect of their existence. Water supports the body, eliminating the need for strong skeletal support. Water provides oxygen, absorbed through gills, thin membranes rich in blood vessels where oxygen diffuses from the water into the blood and carbon dioxide diffuses out. Water facilitates reproduction, allowing eggs and sperm to be released into the environment where fertilization occurs externally, with no need for internal fertilization or parental care. For hundreds of millions of years, vertebrates lived exclusively in water, diversifying into countless forms, from tiny minnows to massive armored placoderms, but always confined to the aquatic realm, unable to survive on land. The transition to land required solving multiple problems simultaneously. How to breathe air, how to support the body against gravity, how to prevent desiccation, how to reproduce without water, and how to sense and navigate in a medium that is 1,000 times less dense than water. The first steps toward land were taken by lobe-finned fish, a group that includes the ancestors of all tetrapods, all four-limbed vertebrates. Lobe-finned fish had fleshy fins supported by bones that correspond to the humerus radius and ulna of tetrapod limbs, and some species had primitive lungs, pouches connected to the throat that could absorb oxygen from air gulped at the surface. These lungs were not efficient enough to support life on land, but they allowed the fish to survive in shallow, stagnant water where oxygen levels were low, giving them an advantage over fish that relied solely on gills. The lobe-finned fish also had strong muscular fins that could support their weight on the bottom of shallow streams, allowing them to push themselves along, perhaps even to drag themselves short distances across mudflats between pools. These adaptations, evolved for life in shallow water, pre-adapted the lobe-finned fish for life on land, providing the raw materials that natural selection would modify and refine over millions of years. The fossil record preserves several transitional forms between fish and tetrapods, fossils that show a mosaic of fish-like and tetrapod-like features, demonstrating that the transition was gradual, not sudden. Tiktaalik, discovered in Arctic Canada and dating to approximately 375 million years ago, had scales, gills, and fins like a fish, but it also had a flat head with eyes on top, a neck that allowed it to move its head independently of its body, sturdy ribs that could support its weight out of water, and fins with bones that correspond to the shoulder, elbow, and wrist of tetrapod limbs. Tiktaalik could probably prop itself up on its fins and lift its head out of the water, perhaps to breathe air or to scan for prey or predators. It represents a stage in the transition from water to land, a creature that was neither fully fish nor fully tetrapod, but something in between, adapted to life in the shallow waters at the edge of the land. Acanthostega and Ichthyostega, two fossils from Greenland dating to approximately 365 million years ago, are even closer to tetrapods. Both had 4 limbs with digits, though Acanthostega had 8 digits on each limb, and Ichthyostega had 7, more than the 5 digits of later tetrapods, indicating that the pentadactyl limb had not yet been established. Both had lungs and gills, indicating that they could breathe both air and water. Both had features that suggest they spent most of their time in water, such as a tail fin and a body shape that was more suited to swimming than to walking. Acanthostega's limbs were probably too weak to support its weight on land, and its digits were more like paddles than feet, suggesting that it used its limbs for swimming or for moving through aquatic vegetation. Ichthyostega had stronger limbs and may have been able to drag itself onto land for short periods, but it was still primarily aquatic. These early tetrapods were not land animals. They were amphibious, living at the interface between water and land, exploiting the resources of both environments, but not yet fully committed to either. The evolution of lungs from the swim bladder, a gas-filled sac that many fish use to control buoyancy, is one of the key innovations that made the transition to land possible. In some fish, the swim bladder is connected to the throat and can be filled with air gulped at the surface, and in some lineages, this connection was modified and the swim bladder became vascularized, rich in blood vessels, allowing it to absorb oxygen from the air. This primitive lung was not efficient, but it was better than nothing, and it allowed fish to survive in oxygen-poor water, and eventually to spend time out of water. Over millions of years, lungs became more complex, more efficient, with increased surface area for gas exchange, with better ventilation mechanisms, with separation of oxygenated and deoxygenated blood, until they could support life on land, where oxygen is abundant but must be actively drawn into the body, not passively absorbed from the surrounding medium. The evolution of limbs from fins is another key innovation, and it required changes not only in the bones but also in the muscles, the nerves, the blood vessels, and the developmental programs that build the limb. The fins of lobe-finned fish had bones and muscles, but they were arranged differently from the limbs of tetrapods, and the transition required the elongation of some bones, the fusion of others, the addition of new joints, the modification of muscle attachments, and the evolution of digits, the fingers and toes that provide grip and leverage on solid surfaces. The genetic basis of limb development is controlled by Hox genes and other regulatory genes that specify the identity of different parts of the limb, and changes in the expression of these genes, in the timing and the location of their activity, can produce dramatic changes in limb structure. The evolution of limbs from fins was not a single mutation, but a series of changes, each one building on the previous one, each one favoured by natural selection, because it provided some advantage, some improvement in the ability to move through shallow water, to push against the bottom, to support a body, to eventually walk on land. The evolution of the amniotic egg, an egg with a shell and internal membranes that protect and nourish the embryo, was a crucial innovation that freed vertebrates from the need to return to water to reproduce. Amphibians, the descendants of the first tetrapods, still lay their eggs in water, and their larvae, tadpoles, are aquatic, with gills and a tail, and they must undergo metamorphosis to transform into the adult form which can live on land. The amniotic egg, which evolved in the ancestors of reptiles, birds, and mammals, contains a series of membranes. The amnion, which surrounds the embryo and contains amniotic fluid, providing aquatic environment for development. The chorion, which surrounds the amnion and facilitates gas exchange. The allantois, which stores waste products. And the yolk sac, which provides nutrients. The shell, which can be leathery or hard, protects the embryo from desiccation and from physical damage, while allowing gas exchange. The amniotic egg allowed vertebrates to reproduce on land without returning to water, and it was a key innovation that enabled the diversification of reptiles, birds, and mammals. The amniotes, which have colonized nearly every terrestrial habitat on Earth, the evolution of the waterproof skin, covered with scales or keratin, was another crucial adaptation for life on land. Amphibians have moist, permeable skin that allows gas exchange but also allows water loss, and they must stay near water or in humid environments to avoid desiccation. Reptiles, birds, and mammals have skin covered with scales, feathers, or hair, and the outer layer of the skin is composed of dead cells filled with keratin, a tough waterproof protein that prevents water loss. This waterproof skin allowed amniotes to colonize dry environments, deserts and grasslands and forests far from water, where amphibians cannot survive. The evolution of waterproof skin required changes in the structure of the skin, in the production of keratin, in the development of glands that produce oils and waxes to further waterproof the surface, and in the behavior of the animal, which must now drink water rather than absorbing it through the skin. The evolution of the three-chambered heart in amphibians And the four-chambered heart in birds and mammals was another important adaptation for life on land. Fish have a two-chambered heart, with one atrium that receives blood from the body and one ventricle that pumps blood to the gills where it is oxygenated, and then to the body. In this system, blood passes through the heart only once per circuit, and the pressure drops as it passes through the gills. So the blood that reaches the body is under low pressure, which is adequate for fish, but not for land animals, which need higher blood pressure to pump blood against gravity, to the head and to the limbs. Amphibians have a three-chambered heart, with two atria and one ventricle, which allows some separation of oxygenated and deoxygenated blood, but there is still mixing in the ventricle, reducing efficiency. Birds and mammals have a four-chambered heart, with two atria and two ventricles, which completely separates oxygenated and deoxygenated blood, allowing higher blood pressure and more efficient oxygen delivery to the tissues, supporting the high metabolic rates of these active, warm-blooded animals. The evolution of the neck, the ability to move the head independently of the body, was another important adaptation for life on land. Fish do not have necks. Their heads are fused to their bodies and they turn by bending the entire body. Tetrapods have necks, with cervical vertebrae that are modified to allow rotation and flexion, giving them the ability to turn their heads to look around, to scan for prey or predators, to reach food, to groom, to interact with others. The evolution of the neck required changes in the vertebrae, in the muscles that move the head, in the nerves and blood vessels that supply the head, and it was a key innovation that improved the sensory and behavioral capabilities of tetrapods, making them more effective predators and more successful survivors on land. The transition from water to land was not a single event, but a series of transitions, each one involving multiple changes, each one taking millions of years, each one driven by natural selection acting on variation, favoring individuals that were slightly better adapted to life at the interface between water and land, and eventually to life fully on land. The fossil record preserves snapshots of this transition, fossils that show the gradual accumulation of terrestrial adaptations, the slow transformation of fish into tetrapods, of aquatic creatures into terrestrial ones, and in the bodies of modern amphibians, reptiles, birds, and mammals, we see the legacy of this transition, the structures and the systems that were modified and refined over hundreds of millions of years, the lungs and the limbs and the amniotic egg and the waterproof skin and the four-chambered heart, all of which trace their origins to the first tentative steps out of the water, to the first breaths of air, to the first footprints on the ancient shore. The silence of the water, the buoyant, weightless existence of the fish, gave way to the harsh reality of land, the pull of gravity, the dryness of the air, the need to support the body, to breathe, to move, to reproduce in an entirely new medium, but life adapted, as it always does, finding solutions, evolving new structures, new functions, new ways of being, transforming the descendants of fish into creatures that could walk and run and climb and fly, creatures that could colonize the land and diversify into the vast array of terrestrial vertebrates we see today. From frogs to lizards to eagles to elephants to humans, all of us carrying within our bodies the traces of that ancient transition, the echoes of the first steps from water to land, the memory of the silence of the ocean, and the first breath of air. The sky was empty for billions of years, a vast expanse of air untouched by life, until approximately 300 million years ago, when the first insects took to the air, and since then flight has been invented independently 4 times in vertebrates, each time using different structures, different mechanisms, different solutions to the same problem of defying gravity. The insects were the first to fly, and they remain the most successful flyers, with more than 1 million described species, many of them capable of aerial acrobatics that surpass anything achieved by vertebrates. Insect wings are not modified limbs. They are outgrowths of the body wall, thin sheets of cuticle supported by veins, hollow tubes that carry air and hemolymph, the insect equivalent of blood. The origin of insect wings is still debated, but they may have evolved from gill-like structures on aquatic insect larvae, or from lateral extensions of the thorax that were initially used for gliding or for thermoregulation, absorbing heat from the sun. Once wings evolved, they were refined by natural selection, becoming larger, more flexible, more efficient, and insects diversified explosively, colonizing the air, exploiting new habitats, dispersing to new habitats. The evolution of flight in insects was a major event in the history of life, and it transformed terrestrial ecosystems, creating new ecological niches, new interactions between organisms, new patterns of diversity. The first vertebrates to fly were the pterosaurs, reptiles that appeared in the late Triassic period, approximately 220 million years ago, and dominated the skies for more than 150 million years until they went extinct at the end of the Cretaceous period. 66 million years ago, along with the non-avian dinosaurs, pterosaurs ranged in size from sparrow-sized species to giants with wingspans exceeding 30 feet, the largest flying animals that have ever lived. The pterosaur wing was a membrane of skin and muscle, stretched from an enormously elongated fourth finger to the body and the hind legs, a structure fundamentally different from the wings of birds and bats. The wing membrane was reinforced with fibers called actinofibrils, which provided strength and flexibility, allowing the wing to change shape during flight, to adjust to different flight conditions, to maneuver, to soar, to dive. Pterosaurs had hollow bones, reducing weight, and they had large brains with well-developed regions for vision and for balance, adaptations for the complex sensory and motor demands of flight. They had diverse diets, some eating fish, some eating insects, some eating small vertebrates, some filter feeding on plankton, and they occupied a wide range of ecological niches, from coastal environments to inland forests to open oceans. The second vertebrate group to evolve flight was the birds, which evolved from small feathered theropod dinosaurs in the Jurassic period, approximately 150 million years ago. The bird wing is supported by the bones of the arm and the hand, which are fused and modified, and it is covered with feathers, complex structures made of keratin, the same protein that makes up hair and scales. Feathers are not simple outgrowths. They are highly organized with a central shaft, the rachis, and branches called barbs, which in turn have smaller branches called barbules, which interlock with tiny hooks, creating a smooth, continuous surface that is both lightweight and strong, capable of generating lift and thrust. Feathers originally evolved for insulation, for keeping the body warm, and they were present in many non-flying dinosaurs, but they were co-opted for flight, modified and refined by natural selection, becoming longer, stiffer, more asymmetric, optimized for aerodynamics. The evolution of flight in birds required not only the evolution of feathers, but also changes in the skeleton, in the muscles, in the respiratory system, in the metabolism, in the nervous system, a suite of adaptations that transformed ground-dwelling dinosaurs into creatures capable of powered flight. The third vertebrate group to evolve flight was the bats, mammals that appeared in the Eocene epoch, approximately 50 million years ago. The bat wing is a membrane of skin stretched between enormously elongated fingers, from the second to the fifth, and extending to the body and the hind legs and often to the tail. The first finger, the thumb, is short and clawed, used for climbing and for grooming. The wing membrane is thin and flexible, rich in blood vessels and nerves, and it can change shape rapidly, allowing bats to maneuver with extraordinary agility, to fly slowly, to hover, to make sharp turns, to catch insects in midair. Bats are the only mammals capable of sustained powered flight, and they are the second most diverse group of mammals after rodents, with more than 1,400 species, occupying a wide range of ecological niches from insectivores to frugivores to nectarores to carnivores to blood feeders. The evolution of flight in bats required changes in the limbs, in the muscles, in the sensory systems, particularly the evolution of echolocation, the ability to navigate and hunt using reflected sound waves, an adaptation that allows bats to fly and hunt in complete darkness. Each of these four groups, insects, pterosaurs, birds, and bats, evolved flight independently, using different structures, different anatomical starting points, different developmental pathways, yet all of them converged on similar solutions to the problems of flight, similar wing shapes, similar flight mechanics, similar adaptations for reducing weight and increasing power. The physics of flight are universal, governed by the same aerodynamic principles, the same forces of lift and drag and thrust and gravity, and natural selection acting on different lineages has produced similar outcomes, demonstrating that there are only so many ways to build a wing, only so many ways to generate lift, only so many ways to fly. The evolution of feathers in birds is one of the most remarkable examples of evolutionary innovation, of a structure that evolved for one function and was later co-opted for another. Feathers are complex hierarchical structures, and their development is controlled by a network of genes that regulate the growth and branching of the feather follicle. The earliest feathers were simple, unbranched filaments, similar to hair, and they provided insulation, trapping air close to the skin, reducing heat loss. Over time, feathers became more complex, with branches and sub-branches creating a larger surface area, improving insulation, and eventually, some feathers became asymmetric, with a longer leading edge and a shorter trailing edge, a shape that generates lift when moved through the air. These asymmetric feathers, the flight feathers, are found on the wings and the tail of birds, and they are essential for flight, providing the lift and the thrust and the control surfaces that allow birds to fly. The evolution of flight feathers from simple insulating feathers is an example of exaptation, the co-option of a structure that evolved for one function for a different function, a common pattern in evolution, where natural selection works with what is available, modifying existing structures for new purposes. The respiratory system of birds is another remarkable adaptation for flight, more efficient than the lungs of mammals, allowing birds to extract more oxygen from each breath, supporting the high metabolic demands of flight. Birds have a system of air sacs, thin-walled structures that extend throughout the body into the bones, and these air sacs act as bellows, moving air through the lungs in a one-way flow from the posterior air sacs through the lungs to the anterior air sacs, and then out of the body. This one-way flow means that the lungs are always filled with fresh air, rich in oxygen, unlike the lungs of mammals, where air flows in and out through the same passages, and the lungs contain a mixture of fresh air and stale air. The one-way flow of air through the bird lung allows for more efficient gas exchange, more oxygen extraction, and it is one of the key adaptations that allows birds to fly at high altitudes, where oxygen levels are low, and to sustain the high metabolic rates required for powered flight. The hollow bones of birds, pterosaurs, and bats are another adaptation for flight, reducing weight without sacrificing strength. The bones are not completely hollow, they are reinforced with internal struts, a structure similar to the trusses in a bridge, providing strength and rigidity while minimizing mass. The reduction in weight is essential for flight, because the power required to fly increases with weight, and every gram saved is a gram that does not have to be lifted, a gram that does not require additional muscle, additional energy, additional metabolic cost. The evolution of hollow bones required changes in bone development, in the deposition of calcium and other minerals, in the remodeling of bone tissue, and it is a common feature of all flying vertebrates, demonstrating that reducing weight is a universal requirement for flight. The evolution of the keel, the enlarged breastbone in birds and bats, is another adaptation for flight. providing a large surface area for the attachment of the flight muscles, the pectoralis major and the supracoracoideus, which power the downstroke and the upstroke of the wing. These muscles are enormous, accounting for up to 30% of the body weight in some birds, and they require a large, strong anchor point, which the keel provides. The keel is present in all flying birds and in bats, and it is absent or reduced in flightless birds like ostriches and penguins, demonstrating that it is specifically an adaptation for flight, not a general feature of birds or mammals. The aerodynamics of flight impose strict constraints on body size and shape, constraints that have shaped the evolution of all flying animals. There is an upper limit to the size of flying animals, determined by the relationship between body mass and wing area, between weight and lift. As an animal gets larger, its weight increases with the cube of its linear dimensions, but its wing area increases only with the square, meaning that larger animals need proportionally larger wings to generate enough lift to support their weight. The largest flying birds alive today, the wandering albatross and the Andean condor, have wingspans of up to 12 feet and weigh up to 30 pounds, near the upper limit for powered flight. The largest pterosaurs, with wingspans exceeding 30 feet, were probably at or beyond the limit for powered flight, and they may have relied primarily on soaring, using rising air currents to stay aloft, flapping only occasionally, conserving energy. The constraints of aerodynamics mean that there will never be a flying animal the size of an elephant, that flight is limited to relatively small animals, animals that can achieve the power-to-weight ratio necessary to generate lift and overcome drag, the migration of birds, the seasonal movement of populations across continents and oceans, is one of the most remarkable phenomena in the natural world, and it is made possible by flight, by the ability to cover vast distances quickly and efficiently. Some birds migrate thousands of miles from breeding grounds in the Arctic to wintering grounds in the tropics, crossing oceans, deserts, and mountain ranges, navigating by the sun, by the stars, by the Earth's magnetic field, by landmarks by instinct encoded in their genes. The Arctic tern holds the record for the longest migration, travelling approximately 44,000 miles per year, from the Arctic to the Antarctic and back, experiencing 2 summers per year, more daylight than any other animal. Migration allows birds to exploit seasonal resources, to breed in the long days and abundant food of the northern summer, and to winter in the milder climates of the south, avoiding the harsh conditions and food scarcity of the northern winter. The evolution of migration required not only the ability to fly long distances, but also the ability to navigate, to orient, to find the way across featureless oceans and unfamiliar landscapes. Abilities that are still not fully understood, but that involve multiple sensory systems and complex neural processing. The evolution of flight has also driven the evolution of predator-prey relationships, of arms races between flying predators and their prey, between flying prey and their predators. Bats and moths have been engaged in an evolutionary arms race for millions of years, with bats evolving echolocation to detect and track moths in the dark, and moths evolving ears that can detect the ultrasonic calls of bats, allowing them to take evasive action, to dive, to spiral, to escape. Some moths have evolved the ability to produce ultrasonic clicks that jam the bat's echolocation, confusing the signal, making it harder for the bat to track the moth. Some moths have evolved scales that absorb sound, reducing the echo return to the bat, making the moth harder to detect. This arms race has driven the evolution of increasingly sophisticated echolocation in bats and increasingly sophisticated defenses in moths, a cycle of adaptation and counter-adaptation that has been ongoing for tens of millions of years and shows no sign of stopping. The evolution of flight has occurred multiple times in multiple lineages using multiple solutions, but it has also been lost multiple times in lineages that became flightless, that returned to a terrestrial or aquatic existence. Flightless birds like ostriches, emus, kiwis, and penguins are descended from flying ancestors, and they have lost the ability to fly, often because they colonized islands where there were no predators and flight was no longer necessary, or because they adapted to a different mode of life, like the penguins, which use their wings for swimming rather than flying. The loss of flight is often accompanied by changes in body size, with flightless birds becoming larger, heavier, more robust, no longer constrained by the need to minimize weight. The loss of flight is also accompanied by changes in the skeleton, with the keel becoming reduced or absent, with the flight muscles becoming smaller, with the wings becoming shorter, sometimes vestigial, remnants of the flying past. The evolution of flight is not just about wings and feathers and hollow bones. It is also about the brain, about the sensory systems, about the nervous system that coordinates the complex movements of flight, that processes the visual information, that maintains balance, that controls the muscles with millisecond precision. Flying animals have large brains relative to their body size, with well-developed regions for vision, for spatial orientation, for motor control, and they have sophisticated sensory systems, including acute vision, sensitive hearing, and in bats, echolocation, the ability to navigate and hunt using sound. The evolution of flight required not only changes in the body, but also changes in the brain, in the nervous system, in the cognitive and sensory capabilities that make flight possible. The sky, once empty, is now filled with life, with insects and birds and bats, each one a testament to the power of natural selection, to the ability of life to adapt, to innovate, to colonize new environments, to exploit new opportunities. The four independent origins of flight, in insects, in pterosaurs, in birds, in bats, demonstrate that flight is not a rare accident, not a fluke, but a solution that evolution has discovered repeatedly, a solution that is favored whenever the benefits of flight, access to new food sources, escape from predators, dispersal to new habitats, outweigh the costs, the energy required to fly, the structural modifications required to support flight, the risks of aerial life. And in the silence of the sky, in the effortless soaring of a hawk, in the darting flight of a bat, in the hovering of a hummingbird, there is a kind of grace, a recognition that life is not bound to the ground, not limited to the water, but can rise, can fly, can claim the air as its own, transforming the empty sky into a realm of life, of movement, of beauty, of endless possibility. You are not entirely Homo sapiens. Within your genome, woven into the very fabric of your DNA, are sequences inherited from other species of humans, cousins who walked the earth tens of thousands of years ago and who mated with your ancestors, leaving traces that persist in you today. For most of human history, we believed that Homo sapiens was the only human species, that we had evolved in isolation, that our lineage was pure, uncontaminated by interbreeding with other hominins. But the sequencing of ancient DNA, extracted from fossils tens of thousands of years old, has revealed a different story, a story of multiple human species coexisting, overlapping in time and space, and occasionally interbreeding, exchanging genes, creating a genetic mosaic that is visible in the genomes of modern humans. The Neanderthals, who lived in Europe and Western Asia from approximately 400,000 years ago until they went extinct around 40,000 years ago, were our closest relatives, sharing a common ancestor with Homo sapiens approximately 600,000 years ago. When the genomes of Neanderthals were sequenced, using DNA extracted from bones found in caves in Europe, scientists discovered that modern humans of non-African ancestry carry approximately 1 to 2% Neanderthal DNA, evidence that Homo sapiens and Neanderthals interbred, probably in the Middle East approximately 50 to 60,000 years ago, when Homo sapiens migrated out of Africa and encountered Neanderthal populations. The Neanderthals were not the brutish primitive creatures of popular imagination. They had brains as large as or larger than those of Homo sapiens. They made sophisticated stone tools. They controlled fire. They buried their dead with grave goods suggesting some form of ritual or belief in an afterlife. They cared for the sick and the injured. And they created art, painting on cave walls, carving symbols into bone and stone. The discovery of a Neanderthal bone flute, a musical instrument carved from the leg bone of a cave bear, demonstrates that Neanderthals had the cognitive capacity for symbolic thought, for music, for culture. They were not inferior to Homo sapiens. They were different, adapted to the cold climates of Ice Age Europe, with stocky bodies, short limbs, large noses and robust skeletons, all adaptations for conserving heat and surviving in harsh environments. The fact that Homo sapiens and Neanderthals interbred, that they produced fertile offspring, that their genes are still present in modern humans, indicates that they were not separate species in the strict biological sense, but rather closely related populations that had diverged but had not yet reached the point of complete reproductive isolation. The Denisovans, another species of human known only from a few fragments of bone and teeth found in a cave in Siberia, also interbred with Homo sapiens. The Denisovan genome, sequenced from a finger bone, revealed that Denisovans were more closely related to Neanderthals than to Homo sapiens, but they were a distinct species, with their own unique genetic variants. Modern humans of Melanesian and Australian ancestry carry approximately 3 to 5% Denisovan DNA, evidence that Homo sapiens encountered and interbred with Denisovans in Southeast Asia or Oceania as they migrated eastward from Africa. Some populations, particularly in Papua New Guinea and Australia, carry even higher percentages of Denisovan DNA, indicating multiple episodes of interbreeding, multiple encounters between Homo sapiens and Denisovans over thousands of years. The genetic legacy of Neanderthals and Denisovans is not evenly distributed across the human genome. Some regions of the genome contain high levels of Neanderthal or Denisovan DNA, while other regions contain none, a pattern that reflects the action of natural selection, favoring some introgressed genes and eliminating others. Some Neanderthal genes have been positively selected, meaning that they provided some advantage to Homo sapiens, and increased in frequency over time. Genes involved in skin and hair, for example, show evidence of positive selection, and it is thought that Neanderthal variants of these genes helped Homo sapiens adapt to the colder climates of Europe and Asia, providing thicker skin, more body hair, better insulation against the cold. Genes involved in the immune system also show evidence of positive selection, and Neanderthal variants of immune genes may have helped Homo sapiens resist diseases that were endemic in Europe and Asia, diseases to which African populations had no prior exposure. Other regions of the genome show evidence of negative selection, meaning that Neanderthal or Denisovan genes were disadvantageous and were eliminated over time. The X chromosome, for example, contains very little Neanderthal DNA, much less than would be expected by chance, suggesting that Neanderthal genes on the X chromosome were harmful, perhaps causing reduced fertility in male hybrids, and were selected against. Regions of the genome involved in brain development and function also contain very little Neanderthal DNA, suggesting that Neanderthal variants of these genes were incompatible with the Homo sapiens genetic background, perhaps causing developmental problems or cognitive deficits, and were eliminated by natural selection. The interbreeding between Homo sapiens and Neanderthals and Denisovans was not a single event, but occurred multiple times in different places at different times, creating a complex pattern of gene flow, of genetic exchange, that is still being unraveled. Some Neanderthal DNA in modern humans comes from early interbreeding events, 50 to 60,000 years ago, while other Neanderthal DNA comes from later events, as recently as 30 to 40,000 years ago. indicating that Homo sapiens and Neanderthals continued to encounter each other, to interact, to interbreed for tens of thousands of years until the Neanderthals finally went extinct. The reasons for the extinction of the Neanderthals are still debated, but it is clear that they were not simply replaced by Homo sapiens. They were absorbed, their genes incorporated into the Homo sapiens gene pool, their legacy preserved in the genomes of billions of people alive today. The discovery of archaic human DNA in modern humans has profound implications for our understanding of human evolution, of what it means to be human. We are not a pure species, not a single lineage, but a mosaic, a mixture of genes from multiple species, a product of hybridization, of interbreeding, of genetic exchange. This is not unique to humans. Hybridization is common in the natural world, particularly in plants and in some animals, and it can be a source of genetic diversity, of new combinations of genes, of adaptive variation that can help populations survive in changing environments. The Neanderthal and Denisovan genes in modern humans are not just relics of the past. They are functional. They are expressed. They affect our biology, our physiology, our susceptibility to disease, our appearance, our behavior. We carry within us the genetic legacy of our extinct cousins, and that legacy shapes who we are in ways both subtle and profound. The EPAS1 gene, which is involved in the response to low oxygen levels, shows evidence of introgression from Denisovans in Tibetan populations. Tibetans live at high altitudes where oxygen levels are low, and they have evolved adaptations that allow them to thrive in this environment, including increased lung capacity, increased breathing rate, and increased production of red blood cells. The Denisovan variant of EPAS1 is associated with lower hemoglobin levels, which may seem counterintuitive, but it actually helps prevent the overproduction of red blood cells, which can lead to thickening of the blood and increased risk of stroke and other cardiovascular problems at high altitude. The Denisovan variant of EPAS1 is found in approximately 80% of Tibetans, but it is rare or absent in other populations, indicating that it was strongly selected for in the Tibetan population, providing a significant advantage in the high-altitude environment. The TBX15 and WA are S2 genes, which are involved in fat distribution and metabolism, show evidence of introgression from Neanderthals in modern humans, and Neanderthal variants of these genes are associated with differences in body fat distribution, with more fat stored in the trunk and less in the limbs, a pattern that may have helped Neanderthals conserve heat in cold climates. Modern humans who carry Neanderthal variants of these genes show similar patterns of fat distribution, demonstrating that these ancient genes are still functional, still affecting our biology, tens of thousands of years after they were introduced into the Homo sapiens gene pool. The OAS genes, which are involved in the immune response to viral infections, show evidence of introgression from Neanderthals in modern humans, and Neanderthal variants of these genes are associated with increased resistance to certain viruses, but also with increased risk of autoimmune diseases, demonstrating that the same genetic variant can have both beneficial and harmful effects, depending on the context, on the environment, on the other genes in the genome. The Neanderthal immune genes may have helped Homo sapiens resist viruses that were endemic in Europe and Asia, but in modern environments, with different pathogens and different lifestyles, these same genes may contribute to autoimmune disorders, to allergies, to inflammatory diseases. The MC1R gene, which is involved in skin and hair pigmentation, shows evidence of introgression from Neanderthals in modern Europeans, and Neanderthal variants of this gene are associated with red hair and pale skin, traits that are common in northern European populations. The Neanderthal variant of MC1R may have been advantageous in the low-light environments of Ice Age Europe, where pale skin allows for more efficient synthesis of vitamin D from the limited sunlight, preventing deficiencies that can lead to bone disorders and other health problems. The presence of this Neanderthal gene in modern Europeans is evidence that some of the physical traits we associate with European populations, traits like red hair and pale skin, may have been inherited from Neanderthals, not evolved independently in Homo sapiens. The study of ancient DNA has also revealed the existence of other, even more ancient, episodes of interbreeding, of gene flow from archaic humans into the ancestors of Neanderthals and Denisovans. The genomes of Neanderthals and Denisovans contain DNA from an unknown archaic human population, a population that diverged from the common ancestor of Homo sapiens, Neanderthals and Denisovans more than 1 million years ago, a population that may have been Homo erectus or another early human species. This ancient DNA is present in very small amounts, less than 1%, but it is detectable, and it indicates that interbreeding between different human species was not a recent phenomenon, not limited to the interactions between Homo sapiens and Neanderthals and Denisovans, but was a recurring pattern throughout human evolution, a pattern that has shaped the genetic diversity of all human populations. The discovery of Homo naledi in South Africa, a species with a mixture of primitive and modern features, small brain, curved fingers adapted for climbing, but also modern-looking feet and legs adapted for walking, has added another layer of complexity to the human family tree. Homo naledi lived as recently as 250,000 years ago, overlapping in time with Homo sapiens, Neanderthals, and Denisovans, and it is possible that Homo naledi also interbred with other human species, though no DNA has yet been recovered from Homo naledi fossils to test this hypothesis. The existence of Homo naledi and of other recently discovered human species like Homo floresiensis, the hobbit from Indonesia, demonstrates that the human family tree was far more diverse, far more complex than we previously imagined, with multiple species coexisting, overlapping, potentially interbreeding, creating a web of relationships that is still being mapped. The human family tree is not a simple linear progression from one species to the next, but a complex branching network with multiple species coexisting, overlapping, interbreeding, exchanging genes, creating a web of relationships that is still being mapped, still being understood. The discovery of archaic human DNA in modern humans has blurred the boundaries between species, has challenged our definitions of what it means to be human, has revealed that we are not separate from our evolutionary past but connected to it, carrying within us the genetic legacy of our ancestors, of our cousins, of the multiple species of humans that have walked the earth over the past 2 million years. The traces of Neanderthals and Denisovans in our genomes are not just scientific curiosities, they are reminders of our shared history, of the deep connections that link all humans, of the fact that we are all related, all descended from a common ancestor, all part of the same evolutionary story. And in the silence of these ancient genes, in the quiet persistence of these genetic echoes, there is a kind of humility, a recognition that we are not the pinnacle of evolution, not the final product, but one branch among many, one species among many that have existed, that have evolved, that have contributed to the diversity of life on Earth. We are a mosaic, a mixture, a hybrid, and that is not a weakness but a strength, a source of diversity, of adaptability, of resilience, a testament to the power of evolution to create, to innovate, to connect, across the vast distances of time, across the boundaries of species, across the entire history of life on Earth. Evolution has not stopped. It continues today in every population, in every generation, in every cell, driven by the same forces that have shaped life for billions of years, mutation and selection, operating on timescales too slow to perceive, but too relentless to ignore. The human species is still evolving, still changing, still adapting to new environments, new challenges, new selective pressures. The lactase persistence gene, which allows adults to digest milk, has evolved independently in multiple human populations within the last 10,000 years, since the domestication of cattle and the adoption of dairy farming. In most mammals, the gene that encodes lactase, the enzyme that breaks down lactose, the sugar in milk, is active only in infants, and it is turned off after weaning because there is no evolutionary advantage to maintaining the ability to digest milk in adulthood when milk is not available. But in human populations that have practiced dairy farming for thousands of years, mutations that keep the lactase gene active throughout life have been strongly selected for because they allow individuals to extract nutrients from milk, providing a significant dietary advantage. The lactase persistence mutation is found in high frequencies in Northern European populations, in some African populations, and in some Middle Eastern populations. But it is rare or absent in populations that have not practiced dairy farming, demonstrating that this is a recent adaptation, a response to a cultural change, the domestication of animals and the consumption of dairy products. The sickle cell allele, a mutation in the gene that encodes hemoglobin, the protein that carries oxygen in red blood cells, provides another example of ongoing evolution in humans. The sickle cell allele causes red blood cells to become misshapen, sickle-shaped, under low oxygen conditions, and individuals who inherit 2 copies of the sickle cell allele, one from each parent, develop sickle cell disease, a serious condition that can cause pain, organ damage, and early death. But individuals who inherit 1 copy of the sickle cell allele and 1 normal copy are resistant to malaria, a parasitic disease transmitted by mosquitoes that kills hundreds of thousands of people each year, primarily in sub-Saharan Africa. The malaria parasite spends part of its life cycle inside red blood cells, and sickle-shaped cells are less hospitable to the parasite, providing protection against infection. The sickle cell allele is found in high frequencies in regions where malaria is endemic, because the benefit of malaria resistance in individuals with 1 copy of the allele outweighs the cost of sickle cell disease in individuals with 2 copies. This is an example of balancing selection, where a harmful allele is maintained in the population because it provides a benefit in certain contexts and certain environments, demonstrating that evolution is not always about perfection, about optimization, but about trade-offs, about compromises, about finding the best solution given the constraints of the environment and the genetic variation available. The FOXP2 gene, which is involved in language and speech, shows evidence of recent positive selection in humans. Mutations in FOXP2 cause severe speech and language impairments, demonstrating that this gene is essential for the development of the neural circuits that control speech. The human version of FOXP2 differs from the chimpanzee version by only 2 amino acids, but these 2 changes occurred relatively recently, within the last few 100,000 years, and they show evidence of strong positive selection, perhaps improving the ability to produce complex speech sounds, to learn language, to communicate with others. The evolution of FOXP2 may have been a key step in the evolution of human language, one of the defining characteristics of our species, demonstrating that even small genetic changes can have profound effects on phenotype, on behavior, on the trajectory of evolution. The future of human evolution is uncertain, shaped by forces that are difficult to predict. In modern industrialized societies, many of the selective pressures that shaped human evolution in the past, such as predation, starvation, and infectious disease, have been reduced or eliminated by technology, by medicine, by agriculture, by sanitation. But new selective pressures have emerged, such as exposure to novel chemicals, to processed foods, to sedentary lifestyles, to urban environments, and these new pressures may be shaping human evolution in ways that are not yet apparent. The pace of cultural evolution, the transmission of information and technology from one generation to the next, has accelerated dramatically in recent centuries, And cultural evolution now proceeds much faster than biological evolution, allowing humans to adapt to new environments, to new challenges, without waiting for genetic changes to occur. But cultural evolution does not replace biological evolution. It interacts with it, creating new selective pressures, new opportunities, new constraints, and the interplay between cultural and biological evolution will shape the future of the human species. Evolution is not limited to humans. It is occurring in all species, all populations, all ecosystems, driven by the same fundamental processes, the same forces of mutation and selection, of drift and migration. Antibiotic resistance in bacteria is one of the most visible examples of ongoing evolution. A direct consequence of the widespread use of antibiotics in medicine and agriculture. When bacteria are exposed to antibiotics, most of them are killed, but a few individuals, those that carry mutations that confer resistance to the antibiotics, survive and reproduce, passing the resistance genes to their offspring. Over time, the frequency of resistance genes in the population increases, and the antibiotic becomes less effective, eventually useless. The evolution of antibiotic resistance is a major public health threat, and it demonstrates the power of natural selection, the speed with which populations can adapt to new selective pressures, the importance of understanding evolution for addressing real-world problems. Pesticide resistance in insects, herbicide resistance in weeds, and drug resistance in viruses and parasites are all examples of ongoing evolution, of populations adapting to human-imposed selective pressures, demonstrating that evolution is not a historical process, not something that happened in the distant past, but a contemporary process happening now in real time in response to the changes we are making to the environment. The evolution of resistance is a challenge, but it is also an opportunity to learn about evolution, to understand the mechanisms of adaptation, to develop strategies for managing resistance, for slowing its spread, for preserving the effectiveness of the tools we use to control pests and diseases. The peppered moth in England provides a classic example of evolution observed in real time, of natural selection acting on visible traits in response to environmental change. Before the Industrial Revolution, the peppered moth existed primarily in a light-coloured form, speckled with dark spots, which provided camouflage against lichen-covered tree bark, protecting the moths from predators. As industrialization progressed and air pollution killed the lichens and darkened the tree bark with soot, a dark-coloured form of the moth caused by a mutation became more common, because dark moths were better camouflaged against the darkened bark, and light moths became more visible to predators. By the mid-20th century, the dark form had become the dominant form in industrial areas, while the light form remained common in rural areas where pollution was low. When air quality improved in the late 20th century, following the implementation of pollution controls, the lichens returned, the tree bark lightened, and the frequency of the light form increased again, demonstrating that evolution can be rapid, reversible, responsive to environmental change, observable within a human lifetime. Climate change is creating new selective pressures on species around the world, forcing them to adapt or go extinct. Some species are shifting their ranges, moving toward the poles or to higher elevations, tracking the climatic conditions to which they are adapted. Some species are changing their phenology, the timing of life cycle events like flowering, migration, and reproduction, to match the changing seasons. Some species are evolving physiological and morphological changes, such as increased heat tolerance, altered body size, or changes in coloration, in response to the new environmental conditions. But not all species can adapt fast enough to keep pace with the rapid rate of climate change, and many are at risk of extinction, unable to migrate, unable to evolve, unable to survive in the new conditions. The current rate of extinction is estimated to be 100 to 1,000 times higher than the background rate, the rate that would occur in the absence of human activity. and we are in the midst of the sixth mass extinction. A loss of biodiversity on a scale not seen since the extinction of the dinosaurs 66 million years ago. The coral reefs, among the most diverse ecosystems on Earth, are particularly vulnerable to climate change, to ocean warming, to ocean acidification, to the bleaching events that occur when corals expel the symbiotic algae that live in their tissues and provide them with nutrients. Some coral species are evolving increased heat tolerance, and some are forming new symbioses with more heat-tolerant algae. But the pace of change is slow, and many reefs are dying, collapsing, losing the diversity of fish and invertebrates that depend on them. The loss of coral reefs would be a catastrophe, not only for the species that live there, but for the millions of people who depend on reefs for food, for coastal protection, for their livelihoods, demonstrating that the impacts of climate change are not abstract, not distant, but immediate, tangible, affecting ecosystems and human communities around the world. The future of life on Earth depends on the choices we make, on the actions we take, on our willingness to recognize that we are part of the web of life, not separate from it, not above it, but connected to it, dependent on it, responsible for it. Evolution will continue, with or without us, shaping life, creating new forms, new adaptations, new species. But the diversity of life, the richness of ecosystems, the beauty and complexity of the natural world are not guaranteed. They are fragile, vulnerable, threatened by habitat destruction, by pollution, by overexploitation, by climate change, by the cumulative impacts of human activity. We have the power to destroy, but we also have the power to protect, to preserve, to restore, to create a future in which humans and nature coexist, in which evolution continues, in which the unbroken chain of life stretching back 4 billion years continues into the future, carrying forward the legacy of all the organisms that have ever lived, all the adaptations that have ever evolved, all the diversity that has ever existed. The story of evolution is not just a story of the past. It is a story of the present and the future, a story that is still being written, still unfolding, still revealing new chapters, new surprises, new wonders. Every fossil discovered, every genome sequenced, every experiment conducted adds to our understanding, fills in gaps, answers questions, raises new questions, deepens our appreciation for the complexity and the beauty of life. Evolution is the unifying principle of biology, the framework that makes sense of the diversity of life, the process that connects all living things, from the smallest bacterium to the largest whale, from the simplest virus to the most complex human, in a single unbroken lineage, a single tree of life, rooted in the deep past, branching into the present, reaching toward the future. The concept of deep time, of the billions of years that separate us from the origin of life, is both humbling and awe-inspiring. If we could compress the entire history of life into a single year, with the origin of life on January 1st, the first multicellular organisms would not appear until late August. The first vertebrates would appear in mid-November. The dinosaurs would appear in mid-December and go extinct on December 26th. And humans would appear only in the last few minutes before midnight on December 31st. Our entire recorded history, all of human civilization, all of the art and science and culture we have created, would occupy only the last few seconds of the year. This perspective, this sense of our place in deep time, is essential for understanding evolution, for understanding that we are not the endpoint, not the goal, not the purpose of evolution, but one branch among many, one species among millions, one moment in a story that spans eons and will continue long after we are gone. And in the silence of deep time, in the patient accumulation of small changes over vast stretches of years, in the relentless motion of evolution, there is a kind of peace, a recognition that we are part of something far larger than ourselves, far older, far more enduring. We are the products of 4 billion years of evolution, the descendants of the first cells that emerged in the ancient ocean. the inheritors of the genetic legacy of countless generations, the beneficiaries of the adaptations that have allowed life to survive, to thrive, to diversify, to colonize every corner of the planet. We carry within us the history of life, written in our DNA, in the structures of our cells, in the rhythms of our metabolism, in the very fact of our existence. And as we drift toward sleep, toward rest, toward the quiet stillness of the night, we can take comfort in knowing that we are part of the unbroken chain, part of the eternal flow of life, part of the story that began in the darkness of the deep ocean and continues today in every heartbeat, in every breath, in every moment of our lives, connecting us to the past, to the present, to the future, to all of life, to the endless, beautiful, relentless motion of evolution.